Good afternoon, everyone. I'm Wiln Jiraldine Novecio, and for today, I'm going to discuss about the Clostridium botulinum. So, this will be the content outline of my presentation. So, I'm going to discuss the physiology and structure, pathogenesis and immunity that talks about its biology and virulence, epidemiology, diseases with clinical cases, and diagnosis that talks about treatment, prevention, and control. So to start my report, let's go first with the introduction. So ang Clostridium are strictly anaerobic to aerotolerant spore-forming bacilli, and they can be they are ubiquitous in the environment and can be found sa kanang mga soil, dust, sa marine and freshwater sediments. Makita po sila sa mga intestinal tract sa animals, like birds ug sa kaning fish. And then sa mga vegetable sad, sa fruits, leaves, silage, ug kani sa mga animal manure. So, kani mga Clostridium are found to grow mo sa kanang mga low oxygen nga conditions. And ang Clostridium botulinum was first discovered and isolated by Emil van Ermingem in 1896 and was later determined to survive by forming spores. So, remaining in a dormant state until mga environmental conditions arise that allow them to grow. So, in 1870, si John Muller, a German physician, derived the name botulism from the Latin word for sausage. So, the bacterium was so called because of its pathological association with the sausages. So, for Latin word for sausage is botulus, and not as it was suggested because of its shape. So, Clostridium botulinum is a bacterium that produces dangerous toxins or botulinum toxins under low oxygen conditions. So, botulinum toxins are one of the most lethal substances known, and these botulinum toxins block the nerve functions and can lead to respiratory and muscular paralysis. And now, let's proceed with the physiology and structure of Clostridium botulinum. So, I have here kanang drawing about the unsay dagway ni Clostridium botulinum, so para mas kanang dali siya nga kanang mahinumduman. So as I've said earlier nga si Clostridium botulinum are gram positive, rod shaped, motile and anaerobic bacteria. So this picture will help you to remember all these points. So as you can see, this is a rod shape. And kanang plus sign sa iyang mata will help you to remember that it is a gram-positive. And then, kanang clip sa iyang ilong signifies that it is anaerobic, meaning they can survive in anoxic environment. And the tire, kanang naa sa iyang tiil, signifies that it is motile. And the spore on the ground will help you to remember that it is a spore-forming bacteria. So here, the figure from the left side is the microscopic view of Clostridium botulinum, and it shows that the shape of Clostridium botulinum is a thin, rod-shaped bacterium. So the size of Clostridium botulinum is about 5 micrometer times 1 micrometer, and the arrangement of cells is arranged singly, in pairs, or in short chains. And they are pleomorphic organisms. Clostridium botulinum is a motile bacterium and flagellated bacterium with peritricate flagella arrangement. The spores are oval, subterminal, and bulging. They are non-capsulated bacterium. And the structure of the spore of Clostridium is also shown here on the right figure. So the genome size of Clostridium botulinum is estimated to be around 4,039 kbp, and the size of chromosome of Clostridium botulinum is relatively larger than many other gram-positive bacteria studied. So why? Because possibly indicating that extra genomic requirements are needed for sporulation or the formation of disease-inducing toxins. So Clostridium botulinum kay maglies siya nga kanang dormant in the form of spores until the right environmental conditions are met. So these spores are very resistant to adverse environmental effects, so making them amenable to most environments and very hard to kill. So the spores will grow under favorable conditions and will begin to produce their toxins as they rapidly propagate. So this figure shows the structure and function of endospores. So endospores mainly resist harsh environments or harsh conditions like high temperatures, disinfectants, radiation, and many more. So endospores are reported to survive for millions of years, and they have this dipicolonic acid and small acid-soluble spore proteins that are crucial in protecting core of the endospore that contains genetic material. So spores also contain dipicolonic acid bound to divalent cations, mostly calcium, so which provides resistance to UV radiation. So small acid-soluble spore proteins bind to the DNA and protect it from heat and chemicals. So small acid-soluble spore proteins also provide nutrients during germination. So, botulism is caused by a group of anaerobic spore-forming organisms called Clostridium botulinum. So, this is classified as a single species but consists of at least 3 genetically distinguishable groups of organisms that have been recognized as toxic for humans. So, they share the ability to produce neurotoxins with similar pharmacological activities but diverse serological properties. So, all clostridial neurotoxins are synthesized as a single inactive polypeptide chain of 150 kDa without a leader sequence and hence are presumably released from the cell by bacterial lysis. So, the toxin types are classified as A, B, C, D, E, F, and G. So, the human botulism has been described with the strains of Clostridium botulinum that produce toxin types A, B, and E. So, less frequently, cases involving type F toxin produced by Clostridium barati and type E toxin produced by Clostridium butyricum and Clostridium argentinense, which is type G toxin, and human diseases only rarely been associated with Clostridium butyricum and Clostridium barati and not definitively demonstrated with Clostridium argentinense. Clostridium botulinum can be divided into 4 subgroups differentiated by their biochemical activities. So, ang group 1 is proteolytic, grows at temperature between 10 and 45 degrees Celsius, and kaning group 1 can produce toxin types A, B, and F. Ang group 2 is non-proteolytic, grows at temperature between 3 and 45 degrees Celsius and can produce toxin types B, E, and F. So those in group 2 are also termed psychotrophic because of their ability to grow at temperatures below 5 degrees Celsius. So groups 1 and 2 are the main groups associated with human illness, and those in group 1 account for almost all reported cases of infant botulism. So group 3 is non-proteolytic and produces toxin types C and D. And group 4 can be weakly proteolytic and produces only toxin type G. Now, let's proceed with the pathogenesis and immunity of Clostridium botulinum. So, Clostridium botulinum is non-invasive. Its pathogenesis is due to the production of powerful neurotoxin, which is called botulinum toxin, probably the most toxic substance known to be lethal to mankind. So, this botulinum toxin is transported via the blood to peripheral cholinergic nerve terminals. And the most common nerve terminal sites are neuromuscular junctions, postganglionic parasympathetic nerve endings, and peripheral ganglia. So, it does not affect the central nervous system. So, in normal conditions, upon stimulation of peripheral and cranial nerves, acetylcholine is normally released from vesicles at the neural side of the motor end plate. So, acetylcholine then binds to specific receptors on the muscle, inducing contraction. Botulinum toxin acts by binding to synaptic vesicles of cholinergic nerves, thereby preventing the release of acetylcholine. So at the peripheral nerve endings, including neuromuscular junctions. So this results in a lack of stimulus to the muscle fibers, irreversible relaxation of the muscles, and flaccid paralysis. So without the release of acetylcholine, the muscle is unable to contract. So what is this acetylcholine? So acetylcholine is a critical neurotransmitter that plays an important role in the normal function of the brain and body. So disruptions in the release and function of this neurotransmitter can result in significant problems in areas such as memory and movement. So because it acts as a chemical messenger to propagate nerve impulses across the neuromuscular junction between a nerve and a muscle. So therefore, botulinum toxin prevents the release of the neurotransmitter acetylcholine, which blocks the neurotransmission at peripheral cholinergic synapses, so leading to a flaccid paralysis. So the only virulence factor is the botulinum toxin. It is a neurotoxin produced by the bacterium Clostridium botulinum. So as you can see here, so this is the normal mechanism sa pag-release sa neurotransmitter, so without the presence of botulinum toxin. So the release of acetylcholine sa kanang neuromuscular junction is mediated by the assembly of the synaptic fusion complex that allows the membrane of the synaptic vesicle containing acetylcholine na mag-fuse siya with the neuronal cell membrane. So the synaptic fusion complex kay set of snare proteins which include synaptobrevin, SNAP25 ug syntaxin. So after sa kanang ma-fuse ang membrane, ang acetylcholine is released into the synaptic cleft and then bound by receptors in the muscle cells sa normal nga neurotransmitter. So here, this figure shows the mechanism of botulinum toxin. So ang kaning botulinum toxin kay magbind siya sa imong neuronal cell membrane at the nerve terminus and then mosulod dayon siya sa neuron by endocytosis. So, ang light chain sa kanang botulinum toxin kay mag-cleave siya sa specific sites sa snare proteins. So, preventing complete assembly of the synaptic fusion complex and thereby yang i-block ang acetylcholine release. So, ang botulinum toxin types B, D, F, ug G kay mag-cleave siya sa synaptobrevin. Tapos, ang type A, C, and E kay mag-cleave siya sa SNAP25. Tapos ang type C kay mag-cleave siya sa syntaxin. So without the release sa kaning acetylcholine, so ang muscle kay dili siya maka-contract. So it will lead to flaccid paralysis. So the botulinum toxins are very similar in structure and function to the tetanus toxin, but differ in their clinical effects kay naa sila gina-target ng lahi nga cells sa nervous system. So, ang botulinum neurotoxins affect the peripheral nervous system, reflecting a preference of the toxin for stimulatory motor neurons at a neuromuscular junction. Tetanus toxin can affect the same system, but the tetanospasmin shows a tropism for inhibitory motor neurons of the central nervous system, and its effects are primarily rigidity and spastic paralysis. So here, it shows the structure of botulinum toxin. So it is synthesized as a single polypeptide chain. And in this form, the toxin has a relatively low potency. The toxin is nicked by a bacterial protease to produce two chains, a light chain or the A fragment and a heavy chain, the B fragment. So as with tetanospasmin, the chains remain connected by a disulfide bond, and the A fragment of the nicked toxin on a molecular weight basis becomes the most potent toxin found in nature. The mechanism by which acetylcholine release is prevented is not known. However, recent evidence suggests that both botulinum toxin as well as tetanus toxin are zinc-dependent endopeptidases that cleave specific proteins that are involved in excretion of neurotransmitters. And both toxins cleave a set of proteins called synaptobrevins. So, synaptobrevins are found in synaptic vesicle of neurons and the vesicles responsible for release of neurotransmitters. So, presumably, Proteolytic cleavage of synaptobrevin 2 would interfere with physical function and release of neurotransmitters. So by contrast, the botulinum indopeptidase inactivates the proteins that regulate release of acetylcholine, blocking neurotransmission at peripheral cholinergic synapses. So because of acetylcholine is required for excitation of muscle, the resulting clinical presentation of botulism is a flaccid paralysis. So, botulism is not transmitted from person to person. The broad category of entry mechanism is passage through intact cells and membranes, which is essential for oral and inhalation poisoning. So in both of these cases, the toxin must cross epithelial barriers to reach the general circulation. And in both cases, the process is described as absorption. Botulism develops if a person ingests the toxin or if the organism grows in the intestines or wounds and toxin is released. So as you can see here, in this picture, botulinum toxin can be absorbed by both the oral route and the inhalation route. So in the case of oral poisoning, either the toxin or the organisms that make the toxin enter the gastrointestinal system. So in the case of inhalation poisoning, only primarily intoxication has been reported. And in both oral and inhalation poisoning, the toxin binds to the apical surface of epithelial cells, after which it is carried to the basal surface of cells and released into the general circulation. So for short, botulinum toxins are absorbed from the intestinal tract or the infected wound site and are carried via the lymphatic system and from the intestinal tract by the bloodstream to the neuromuscular endings. So Toxin types differ in their affinity for nerve tissue. So with type A having the greatest affinity, and the toxin must enter the nerve ending to exert its effect. Now let's proceed with the epidemiology of Clostridium botulinum. So Clostridium botulinum, as I've said earlier, is commonly isolated in soil and water samples throughout the world. So in the United States, ang type A strains are found mainly in neutral or alkaline soil west of the Mississippi River. The type B strains are found primarily in the eastern part of the country in rich organic soil, and type E strains are found only in wet soil. Although Clostridium botulinum is commonly found in soil, disease is uncommon in the United States. And there are a total of 177 cases, including 137 cases of infant botulism were reported in 2017. So, according ni siya sa katong CDC. So, there are three main types of botulism, namely foodborne, infant, and wound. And there are another two clinical categories are recognized as adult intestinal toxemia and iatrogenic botulism. So first, let's talk about the foodborne botulism. So it is the most common form of botulism and it is caused from ingestion of foods containing Clostridium botulinum toxin. So in the case of foodborne botulism, contaminated products may be widely consumed, thus exposing a large number of people to the hazard and may subsequently represent a medical and public health emergency. So the common sources sa kaning foodborne botulism are kanang mga homemade nga foods nga wala na properly na canned or na preserve or na ferment. Though uncommon siya, store-bought foods also can be contaminated with botulinum toxin katong mga processed nga foods. So foodborne botulism is not an infection but an intoxication since it results from the ingestion of foods that contain the preformed clostridial toxin. So the botulinum toxin is ingested with food in which spores have germinated and the organism has grown. So the toxin is absorbed by the upper part of the gastrointestinal tract in the duodenum and jejunum and passes into the bloodstream by which it reaches the peripheral neuromuscular synapses. So the toxin binds to the presynaptic stimulatory terminals and blocks the release of neurotransmitter acetylcholine, which is required for a nerve to stimulate the muscle. So the responsible toxin for foodborne botulism is type A, B, and E toxins. So kani nga mga toxin does not give a bad odor or taste to food. An average of 110 cases of botulism is reported annually in the United States, and about 25% of these cases are foodborne botulism. So, infant botulism affects infants less than 1 year of age and is caused when infants ingest Clostridium botulinum spores, so which then germinate and produce toxin in the intestine. So the spores may come from the environment or from foodstuffs. So a survey of foods commonly fed to infants revealed that Clostridium botulinum in specimens of corn syrup as well as honey. So honey has been previously implicated in the disease and is no longer recommended for infants less than 12 months. So almost all cases of infant botulism have been due to types A, B, E, and F. So this is a result of colonization of the intestinal tract after ingestion of spores of Clostridium botulinum. So the infant intestinal tract often lacks both the protective bacterial flora and the Clostridium inhibiting bile acids found in normal adult intestinal tract. So honey was found to be the vehicle of the spores in 26 cases, and most cases occur before the age of 6 months. And the microbiological surveys of honey products have reported the presence of Clostridial spores in up to 25% of products. So for this reason, honey should not be given to children during the first year of life. An average of 110 cases of botulism is reported annually in the United States, and approximately 70% of these cases are infant botulism. So, wound botulism occurs when Clostridium botulinum infects a wound and subsequently produces toxin, and it has been associated with major soil contamination through compound fractures or severe traumas or lacerations. So, the incidence of wound botulism has increased considerably in recent years, particularly among injectors of black tar heroin. An average of 110 cases of botulism is reported annually in the United States. And over the past few years, the number of cases of wound botulism has been between 30 and 40%. So the other types of botulism is the adult intestinal toxemia and iatrogenic botulism. So the adult intestinal toxemia is the colonization of the intestine with Clostridium botulinum following ingestion of spores and is analogous to infant botulism. Germination, vegetation, and toxin production by Clostridium botulinum are not usually supported in the normal adult intestine. However, in a small number of adults who have a bowel abnormality or bowel disease or are undergoing antimicrobial therapy, colonization can occur. So adult intestinal toxemia, are also known as adult intestinal colonization. Botulism is a very rare kind of botulism that can happen if the spores of the bacteria get into an adult's intestines, grow and produce the toxin. So similar to infant botulism. Although we don't know why people get this kind of botulism, people who have serious health conditions that affect the gut may be more likely to get sick. And the iatrogenic botulism is the accidental overdose of toxin via inhalation by laboratory workers or via injection for therapeutic or cosmetic purposes. So iatrogenic botulism is the most recent man-made form of botulism, which is a rare acquired neuromuscular junction disease with descending flaccid paralysis caused by botulinum neurotoxins, and it may occur as an adverse event after therapeutic or cosmetic use. So iatrogenic botulism can happen if too much botulinum toxin is injected for cosmetic reasons, so such as for wrinkles, or medical reasons, such as for migraine headaches. So now we know about the pathogenesis of botulism, and now let's talk about some of the clinical features. So botulism infections have a varied symptomatology, so they have anywhere from mild symptoms to severe and life-threatening symptoms, and their incubation period, depending on how you become infected, varies as well. So incubation period vary depending on the type of botulism infection. So we're gonna talk about the 3 types of botulism infections, but all 3 types of botulism infections have characteristic findings. So they all have findings that are in common. One is that they all have symmetric descending paralysis. So it's on both sides of the body, and it starts from the head and goes down toward the toes. Second one is that they don't have a fever. The third is that they may have visual deficits, they may have issues with sight, and they don't have other sensory deficits. So again, a very key finding with botulism is that there are no other sensory deficits. The fourth is that patient, although they have symmetric descending paralysis, they are alert and responsive. They know what's going on, so they're not unresponsive in crushing. They are actually alert and responsive. And lastly, looking at their vital signs, we see normal blood pressure and maybe a slow to normal heart rate, but again, their vital signs are normal. So first, let's talk about infant botulism, or also known as fluffy baby syndrome. So decreased or no muscle tone, or medically termed as hypotonia, so which is related to that characteristic finding of asymmetric descending paralysis. So infant botulism is a sporadic disease. While it is a serious illness for the affected child, it does not pose a public health threat because it is extremely rare and has no epidemic potential. So it affects infants from the age of 1 week to 12 months old, but mostly under the age of 6 months. So all of this is because of the nature of immature gut flora in an infant, so the infant has not developed a mature gut microbiome or gut flora. So the instances, if they were to eat or consume dust that have Clostridium botulinum spores, those spores can enter into their intestinal system, and those spores can then form into the Clostridium botulinum bacteria. And those bacteria can flourish, and this can multiply in a layer of the intestinal mucosa, and they can produce toxins that cause infant botulism. So the toxins responsible for infant botulism are toxins A, B, E, and F. And infant botulism is classically associated with eating honey. Okay, so I have here a clinical case of infant botulism reported by Centers for Disease Control and Prevention. So this case is a 10-week-old infant with a history of constipation in the first month of life was admitted to a hospital after having difficulty in sucking and swallowing for 2 days. So the infant was irritable and had loss of facial expression, generalized muscle weakness, and constipation. Mechanical ventilation was required for 10 days because of respiratory failure. A diagnosis of infant botulism was established 29 days after onset of symptoms by detection of Clostridium botulinum producing toxin type B in stool enrichment cultures. So the patient was treated with botulism immune globulin intravenous or BIG4 and discharged fully recovered after 20 days. So in contrast with foodborne botulism, diagnosis of infant botulism can be made by detecting the organism in the baby's stool. So here, the disease typically affects infants younger than 1 year, and the symptoms are initially non-specific. So progressive disease with flaccid paralysis and respiratory arrest can develop. However, mortality in documented cases of infant botulism is very low, and some infant deaths attributed to other conditions like sudden infant death syndrome may actually be caused by botulism. So next is the foodborne botulism. So it is often caused by eating canned foods that have not been canned properly and that contains the toxin. So what happens here is that after the ingestion of the toxin from the food, the incubation period of this condition is anywhere from 10 to 35 hours. So it mostly caused by toxins A, B, and E. So prodromal symptoms with regards to nausea, vomiting, diarrhea, dry mouth, and sore throat. And once we get the symptoms of food botulism, these include vertigo, dry eyes and blurry visions, ptosis with dilated fixed pupils. So there is a clinical case reported by the Centers for Disease Control and Prevention about the foodborne botulism. So it caused an outbreak associated with contaminated carrot juice. So the patients with foodborne botulism typically become weak and dizzy 1 to 3 days after consuming the contaminated food. So initial signs include blurred vision with fixed dilated pupils, dry mouth, constipation, and abdominal pain. So fever is absent. Bilateral descending weakness of the peripheral muscles develops in patients with progressive disease or flaccid paralysis, and death is most commonly attributed to respiratory paralysis. So patients maintain a clear sensorium throughout the disease. Despite aggressive management of the patient's condition, the disease may continue to progress because the neurotoxin is irreversibly bound and inhibits the release of excitatory neurotransmitters for a prolonged period. So complete recovery in patients often requires many months to years, or until the affected nerve endings regrow. Mortality in patients with foodborne botulism, which once approached is 70%, has been reduced to 5% to 10% through the use of better supportive care, particularly in the management of respiratory complications. So the third type is wound botulism. So it has prolonged incubation period, usually around 10 days. So what happens here is that ang spores mosulod siya sa imong open wound. So example, if ang soil has spores and then the spores can get into the wound, but kani siya is rare lang, kaning wound botulism. So it is similar to foodborne botulism except there are no prodromal gastrointestinal symptoms because the spores are not making it into the gastrointestinal tract. So we shouldn't have gastrointestinal symptoms. But it may have fever and leukocytosis or increase in white blood cell count. So this fever is more likely due to a secondary infection and not because of the spores, but maybe from another type of bacteria entering the wound causing a fever. So everything else seemed to be very similar to the foodborne botulism. So for short, wound botulism develops from toxin production by Clostridium botulinum in contaminated wounds. Although the symptoms of disease are identical to those of foodborne disease, the incubation period is generally longer, 4 days or more, and the gastrointestinal tract symptoms are less prominent. And now let's proceed with the clinical diseases of Clostridium botulinum. So the clinical diagnosis of foodborne botulism is confirmed if toxin activity is demonstrated in the implicated food or in the patient's serum, feces, or gastric fluid. So infant botulism is confirmed if toxin is detected in the infant's feces or serum or the organism cultured from feces. So wound botulism is confirmed if toxin is detected in the patient's serum or wound or if the organism is cultured from the wound. Toxin activity is most likely to be found early in the disease, and no single test for foodborne botulism has sensitivity greater than 60%. So in contrast, toxin is detected in the serum of more than 90% of infants with botulism. And there are various culture media used for the cultivation of Clostridium botulinum in the laboratory, and most commonly the sheep blood agar medium and CMB medium is used. So the other media are nutrient agar medium, Columbia horse blood agar medium, alkaline glucose gelatin medium, egg yolk agar medium, and Clostridium botulinum isolation or CBI agar medium, which is a selective medium. So in sheep's blood agar medium, it is differential media because 3 different types of hemolysis or lysing of red blood cells can be seen on this plate. So Clostridium botulinum grows well, and many strains of Clostridium botulinum show beta hemolysis or the complete hemolysis characterized by a clear zone around the colonies, which is caused by the production of botulinolysin, which is a hemolysin toxin. However, there are many strains that do not produce this toxin, hence shows no hemolytic activity in the blood agar medium. So in Clostridium botulinum isolation agar, which is a selective medium for Clostridium botulinum, the colonies are similar as that on egg yolk agar, but it prevents the growth of contaminant and only allows the growth of Clostridium species, especially Clostridium botulinum, and commonly employed for its isolation from fecal matter, where a large number of contaminant bacteria are present. So this medium contains cycloserine, sulfamethoxazole, and trimethoprim as selective inhibitory agents. So this figure is the cultural characteristics of Clostridium botulinum on different media. Also, isolation of Clostridium botulinum from specimens contaminated with other organisms, so example, feces, wounds, can be improved by heating the specimen for 10 minutes at 80 degrees Celsius to kill all non-spore-forming bacteria. So, culture of the heated specimen on nutritionally enriched anaerobic media allows the heat-resistant Clostridium botulinum spores to germinate. So, demonstration of toxin production typically performed at public health laboratories must be done with a mouse bioassay. So this procedure consists of the preparation of 2 aliquots of the isolate, mixing of 1 aliquot with antitoxin, and intraperitoneal inoculation of each aliquot into mice. So if the antitoxin treatment protects the mice, toxin activity is confirmed. So samples of the implicated food, stool, specimen, and patient's serum should also be tested for toxin activity. Lastly, let's talk about the treatment, prevention, and control of Clostridium botulinum. So, botulism is caused by a toxin that attacks the body's nerves and causes difficulty in breathing, muscle paralysis, and even death. So, doctors treat botulism with a drug called an antitoxin, which prevents the toxin from causing any more harm. So, antitoxin does not heal the damage the toxin has already done. So, depending on how severe your symptoms are, you may need to stay in the hospital for weeks or even months before you are well enough to go home. If your disease is severe, you may have breathing problems. So, you may even have respiratory or breathing failure if the toxin paralyzes the muscles involved in breathing. So if that happens, your doctor may put you on a breathing machine or ventilator until you can breathe on your own. So the paralysis caused by the toxin usually improves slowly. The medical and nursing care you receive in the hospital is to help you recover. People with wound botulism sometimes need surgery to remove the source of the bacteria and may need to take antibiotics. Patients with botulism requires the following treatment measures. So, like adequate ventilatory support, elimination of the organisms from the gastrointestinal tract through the judicious use of gastric lavage and metronidazole or penicillin therapy, and use of trivalent botulinum antitoxin versus toxins A, B, and E to inactivate unbound toxins circulating in the bloodstream. Ventilatory support is extremely important in reducing mortality, and protective levels of antibodies do not develop after disease, so patients remain susceptible to botulism. So the disease is prevented by destroying the spores in food, preventing spore germination by maintaining the food in an acid pH or storage at 4 degrees Celsius or colder, or destroying the preformed toxin, or all botulinum toxins are inactivated by heating at 60 degrees Celsius to 100 degrees Celsius for 10 minutes. So spore germination in foods prevented by maintaining food at an acid pH by high sugar content, so example, fruit preserves, or by storing the foods at 4 degrees Celsius or colder. So infant botulism has been associated with consumption of honey contaminated with Clostridium botulinum spores, so children younger than 1 year should not eat honey. So I guess that ends my presentation. So I hope you learned something, though lengthy kayo akong report. And thank you and stay safe everyone.